Hello friends, welcome to Medico's Ninja. In this video we will be talking about the chapter culture media. In culture media we will study about the bacteria or the microorganisms grown in a culture, that is in a petri dish or any test tubes. Before studying the types of culture media, we will study about the constituents of culture media. Culture media consists of water, electrolyte, peptone, meat extract, yeast extract, blood, and agar. Water is a normal constituent of hydrogen and oxygen. Electrolyte is the sodium chloride and etc. Next is peptone. Peptone are the partially digested proteins produced from lean meat. The composition of peptones are proteose, amino acid, polypeptide, phosphate, and minerals. Next is meat extract, yeast extract, blood, and agar. Meat extract available as lab lemco. Small pieces of meat are present in this culture media, and next is yeast in this culture media. To see the microorganisms, we use the yeast extract prepared from the baker's yeast. Next is blood or serum, and next is the agar. Agar is prepared from the seaweed. Contains mainly long chain polysaccharide. It can be in the powder form or in the shred forms. Types. Next, studying about the types of culture media. Culture media can be classified on the basis of the physical state, solid, semi-solid, liquid. On the basis of presence of molecular oxygen, aerobic and anaerobic. On the basis of nutritional factors, simple, complex, synthetic, special. Simple can be so. On how many bases did we divide the culture media? Culture media was divided on the basis of physical state, molecular oxygen, and nutritional factors. Physical state, solid, semi-solid, and liquid media, while molecular oxygen, aerobic and anaerobic. In aerobic, oxygen is required, while in anaerobic, no oxygen is required for the culture media. Next, on the basis of nutritional factors classification of culture media can be done on the basis of can be done as follows: simple culture media, simple media, complex media, synthetic media, special media. In simple media, we use example is nutrient broth and the glucose broth. In each nutrient broth consists of peptone water. Each nutrient broth consists of peptone water and meat extract. Peptone water and meat extract. While in glucose broth there occurs the presence of peptone water along with the glucose. Next is the complex media. In complex media can be categorized as the media used other than simple media are the complex media. Next is the synthetic media. Synthetic media is derived from pure chemicals. And while next is the special media. In special media can further be classified into various categories: enrichment media, selective media, differential media, indicator media, and sugar media. In enriched media, first we will be studying about the enriched media. In enriched media, as the word signifies, the enriched particles can be used in this type of media, such as blood, serum, egg, etc. For the study of enriched bacteria, we will use the blood agar, chocolate agar, and the Loeffler's serum slope. In the blood agar, further we will all be studying about this. Now just we'll go through it. Next is enrichment media. In enrichment media, we use the substances which are used for the enrichment enrichment of any culture media. This consists of various examples such as tetrathionate broth, selenite broth, alkaline peptone water. Tetrathionate broth, selenite broth, and alkaline peptone water. Next is the selective media. So in selective media, a selective type of bacteria or inoculum, selective type of substances are used which either facilitate or inhibit the culture media. In selective media, deoxycholate, citrate agar, bile salt agar can be used. Next is differential media. In differential media, those substances are used which are helpful in differentiating the characteristics of bacteria. Example is McConkey's agar. Next is indicator media. In indicator media, these media are used to indicate these, as the word signifies, these media are used only for the indication purpose. In this, there occurs the example of McConkey agar, blood agar. In McConkey agar, where have we studied? We have studied McConkey agar in differential media, while a blood agar is studied under the enriched media. Next is sugar media. In sugar media, we will study about the Durham's tube, where there occurs the gas production. Earlier we have said that there will be the description of these different special type of culture media. First is enriched media. In enriched media, we will be talking about the blood agar, chocolate agar, and Loeffler's serum Loeffler's agar. In blood agar, there occurs the culture of blood, so there will be the red culture. In this we will see the growth of streptococcus, while in next is the chocolate agar is brown or black in color. In this also we will study about the blood culture, but the only difference between the blood agar and chocolate agar is that in blood agar there occurs only the culture of blood, while in chocolate agar there occurs the heating of blood, then after the culturing. In this chocolate agar can be used to study the Neisseria and Haemophilus influenzae. Next is the serum Loeffler's agar. Serum Loeffler's agar are used for the isolation of Corynebacterium diphtheria. Can be used for the isolation of Corynebacterium diphtheria. In next type of special media is enrichment media. This enrichment media can also be used into different types. Tetrathionate broth, selenite F broth, and alkaline peptone water. Tetrathionate broth inhibit the coliform and typhoid paratyphoid bacilli can grow. While in salinite broth, next talking about salinite broth is also similar to the tetrathionate broth. जैसे इसमें टाइफॉइड पैराटाइफॉइड बैसिलाई के ग्रोथ को देखते हैं, वैसे ही इसमें भी ग्रोथ को ही देखते हैं. नेक्स्ट इज अल्कलाइन पेप्टोन वाटर. इन दिस वी स्टडी अबाउट द विब्रियो कॉलेरी ग्रोथ. अल्कलाइन पेप्टोन वाटर वी सी अबाउट द विब्रियो कॉलेरी ग्रोथ. Next type of media is the selective media. In selective media, we have earlier studied that in this type of media we see or study about the specific culture media only. Some of the examples of selective media are deoxycholate citrate agar, bile salt agar, sodium potassium telluride blood agar, and Wilson Blair medium. Two important what I think are deoxycholate citrate agar and bile salt agar. Deoxycholate citrate agar can be used for the stool examination in any of the diarrhea type of diseases. Deoxycholate citrate agar can also be used. Next is bile salt agar. In bile salt agar also we will see the vibrio cholerae growth. In alkaline peptone water, which act as an enrichment media, also in there also we will see the vibrio cholerae agar. While in selective media, bile salt agar selective media, here also we will see the vibrio cholerae growth. Next is potassium telluride blood agar and Wilson Blair medium. You can study about this in the book. Next we will be talking about the differential media. In differential media, when a medium contains substances which helps to distinguish different characteristics of the bacteria are the differential media. In this differential media, we will be studying about the McConkey medium and cysteine lactose electrolyte deficient agar. McConkey medium is an important one, but the alternative for McConkey medium is cysteine lactose electrolyte deficient agar. It can also be called as CLED agar. Cysteine lactose electrolyte deficient agar. Next we will be talking about the indicator media. In indicator media, as the word signifies, this culture media can act as an indicator whether the growth is possible in it or not. We can see with this Indicator media, there are various types of indicator media. One of them are McConkey agar, and some of them are McConkey agar while blood agar. McConkey agar, as we have studied, is used in differential media, while next is blood agar. Blood agar is used in selective media, no, sorry, enriched media. Next, we will be talking about the transport media. As the word signifies, this media is used for the transport of uh, this media is used for the transport of bacteria from one media to another. For this transport media, there occurs two types of media: VR, Venkatraman fluid, Venkatraman Ramakrishnan fluid, or VR fluid, and Stewart transport media. Next type of media is sugar media. Sugar media, as the word signifies here also, sugar presence of sugar is important. In this, we take a large tube, and a small Durham's tube is inserted into the large test tube. This, and we will observe the gas production. If there occurs the gas production, then this sugar media, and then we can say that the bacteria has grown into this culture, this type of sugar culture media. And this whole what we have talked about are the aerobic type of culture media. In while anaerobic culture media can be classified into two types: cooked meat broth, and next is thioglycolate broth. Thank you for this, for seeing my video. And if you like my videos, then you can like and subscribe. We'll be talking about next many things about in the next chapter. Till then, take care. Bye bye. Thank you.